Well, thanks, Tom, for those nice remarks, some of which were true. It's a delight to be here. It's a real honor to be part of this, I guess, annual event. Certainly in such a place with the tradition that you have here at CSU in ecology, to be recognized here is really quite an honor. So I'm delighted to be here. I've enjoyed the discussions and look forward to more of them over the next day or so. So what I'm going to do today is talk about some of the ways we've been thinking about responses to climate change. And I think a lot of us are really sort of interested in climate effects at this scale. We want to know how distribution and abundance is going to be affected by a warmer climate in the southeast. We're confronted with predictions that things are going to warm up and the summers or the winters will look more like the Caribbean perhaps, but the summers might be something quite different. So these are novel. These are novel conditions, novel combinations of environments. We can't go anywhere in the landscape today and find the kinds of situations we're confronted with in the future. Over a decade ago, I was involved in trying to predict spread of species in response to climate change, thinking about how species had responded to big climate changes in the past, things like glacial to interglacial cycles. And one of the things that really struck me, and this is right out of some of our papers from over a decade ago, is really a couple of things. First of all, I think there was a pointer up here. Was that true or did I imagine it? Oh, thank you. One of the things that came out of this work that was impressive to me was first of all the fact that the kinds of projections you might get for spread based on fitted models were really not that helpful. You got values all the way from a few meters to kilometers per year for most of the species we looked at. And it wasn't the case that you could collect more data and know more, because if you knew parameters precisely, it wouldn't help the predictions any. So in other words, it didn't help to go out and collect more and more dispersal data if you wanted to anticipate changes in distributions with climate. It had to do with the process itself. So that was interesting. It wasn't going to be helpful just to go out and collect more and more data. And that's something that a lot of people were trying to do at the time, is to say, well, we'll collect long distance dispersal data, we'll fit models, and then we'll be able to predict. The second is that really a lot of the estimates that we could come up when we fit real data to these models were actually quite slow. So here we're down in less than tens of meters per year. Certainly quite a bit slower than would be needed to keep up with contemporary climate change. Well, more recently with Kai Zhu, Chris Woodall, we looked at the FIA data, and these are interesting because we have both trees and seedlings. And any population model of spread essentially predicts that you've got this leading front of seedlings, juveniles, out in front of the main population. So we said, well, let's just look at this. Let's look at where the seedlings are relative to where the adults are. And so if you plot the amount of climate change, which varies geographically through time, the southeast, not much change, quite a bit in the northeast and upper Midwest. What we find is that contrary to what you would predict, lots of climate change, lots of spread, we actually found an awful lot of species that really look like they're contracting. Now, there's a whole lot of reasons why you could get either pattern. But on the other hand, 40,000 plots is a pretty good sample. So you know, this doesn't mean that nothing's responding to climate change. But what it does say is we're not seeing nearly the type of pattern that we would expect to find if these populations were in fact tracking climate change. We're not seeing this sort of positive relationship between the two. This seems to have a mind of its own, which is probably good. Anyway, back at this time, one of the things I got into was trying to understand better how these processes operate at the scales of individuals. and thinking about the fact that when we aggregate all the individuals together and look at what a species is doing, it could be a very different story than what we would get if we looked at the individuals in aggregated seconds. So in other words, you can aggregate then analyze, or you can analyze then aggregate. So I've really focused on this first approach to really try to understand what's happening at the individual level, and then try to say what that can help us with at the species level. So in terms of coexistence, I think you can make a pretty strong case that this is really a good way to go. Theory tells us in order for species that compete to coexist, we need some sort of trade-offs between responses. Let's say we might have two species responding to two niche axes. You need some sort of way for one to win when the other is doing poorly and vice versa. This means that everybody's got a chance. But when we look at observations, we often find the case where when it's good for one species, it's good for another species. A lot of species in eastern deciduous forests, when it's warm and wet, that's good. When it's cool and dry, that's bad. So you tend to, at the species level, get these very positive correlations. So certainly that's not consistent with the kind of pattern that we would expect for coexistence to occur. Let's disaggregate. So if we go to the scale of individuals and ask how closely correlated are two individuals, what we find is really the opposite case. If you look at the correlations between individuals of the same species, you tend to get much higher correlations than between individuals of different species. Well, really, this is all you need to promote coexistence. In other words, you need some sort of process where individuals of the same species concentrate competition within the species because they're responding more alike. Now, this can accumulate over time, spatially, and so when I looked at sort of the distribution of the fraction where correlations within a species are greater than between species, these correlations are quite high for many, many species. At the species level, they all look positively correlated. At the individual level, they're acting more like others of the same species. And so again, when you start to think about how this plays out, that's a way to concentrate competition within the species, despite the fact that many of the species level correlations are going to be positive. So now if we start to apply this approach to climate, I think there's a lot of things we can do when we start to think about how individuals respond to variation in climate. And so the first thing to sort of think about is it's not the species responding to climate, it's the individuals responding to weather. That's where the process happens. Everything else is an aggregate. And so if we look at, for example, water use by individual trees, you can see very different responses of individuals. These happen to be different species. They depend on which species it is, but nonetheless at the individual scale you can see a lot of dynamics going on. So we want to sort of try to get from that scale up to the scale where these species level interactions occur. So what I want to do today is ask really, do these individual scale processes matter for the aggregate response to climate? And I'll say that the answer is yes. And what I want to do is sort of quantify these individual scale interactions in terms of how individuals respond to different climate variables, but also how they are controlled by interactions with things like competition. And that will bring us down to this reason why we might want to think about how this individual scale is really critical for the aggregate. Just to sort of put this in context of some of the ways that we currently are modeling responses to climate, I think these are all good approaches, but there's reasons why we might want to supplement them with individual scale data. I'll mention a few things about species distribution modeling, DGVMs, and also just large-scale multi-factor global change experiments. Well, the big models that we use really have to admit this process level scale. And even when we've got FIA data, which include, at least in the East, we have repeated censuses, a few repeated censuses on a number of plots. We've got very few input variables for those, and they contain very little demographic information. There's only 5 years apart, so you don't know annual climate dependencies, and so there's not a lot we can do with them, and they don't really get at this scale. When we take dynamic simulations, we've also been parameterizing these with species level parameters. And just to sort of try to illustrate the problem when we use aggregate variables, and these are recognized by various names in different disciplines, the ecological fallacy, numbers of terms that are used for this. This is essentially the problem you get if you have a couple of species, let's say red and black, are responding along 2 axes. If we just take marginal parameter values, which are shown along the margins, here's an example where black wins in both cases. So in other words, it's got a bigger response on this axis, it's got a bigger response on this axis. Here is the joint distribution that it's describing. Well, if individuals are competing, this marginal distribution is not the relevant response. It's what's happening to individuals, which is a conditional distribution. So in other words, the response of an individual is conditioned on the environment it sees, not the marginal for the population. When you compare the aggregate response to the conditional response, it flip-flops. And so you actually get the opposite result. So this is just a clear case of the problems you get when you start parameterizing simulation models with aggregate variables, is you miss the dynamics that occur when things are interacting. And as we all know, ecosystems involve a very rich set of interactions. So we've all recognized how complicated these interactions can get. Here's a nice summary from Frelich and Reich a few years ago. Just thinking about these responses in the upper Midwest and all the different ways that these interactions can occur. And one of the things they point out here is that wet sites might provide a refuge for species that require moist conditions. Well, this is an example of a negative interaction because it means that the biggest effect of precipitation would be on sites with the lowest soil moisture. So that's negative, right? So if the biggest response to one variable occurs where another variable is low, that's a negative interaction. And so you could sort of think about classifying this way. So this is just a little cartoon where I've sort of mapped out, here's a positive and a negative interaction to 2 variables. The surface could be something like growth rate, it could be fecundity, it could be survival probability, some response variable that we care about. A positive interaction means that the biggest response to one variable is occurring where that variable is high, and a negative interaction is just the opposite. And one way you can kind of see this is that the fat arrows with the big response is crossing a lot of contours. So that's how you can sort of read a map like this. So there's a positive, there's a negative interaction. One of the reasons why it's hard to interpret interactions is you often get situations like this, and this might not actually be biologically sensible, but that's what you get. So that's what happens with interactions in observational data. Now, if you have a multi-factor experiment, this is also a challenge too. And so think about an interaction, and we're going to manipulate 2 variables. And again, we'll think about some response described by a surface here. And we might start the initial control treatment as a white box, and maybe we'll increase one variable and decrease another. Maybe that's our experiment. So we'll start at a white box and we'll end up at a red box. Well, if we've got a positive interaction like this, we'll see the biggest response to the first variable up here, whereas if we're down here, we'll see the biggest response to the second variable. So we don't know what this surface looks like, but we see very different reactions depending on where we start from. Now you can also get different responses if it's a negative interaction. Now if we don't know where we are on this surface, this experiment can give us any sort of combination of responses. So in order to really understand what's going on, we need to know this surface. We need to figure out the surface. So multi-factor experiments where we've got a treatment, a one-way or a two-way interaction, three-way interactions, if we don't really have enough variation, which we never really do, you know, think of phase experiments, our own warming experiment, you know, I'm the guilty one in all of this. We do a lot of this sort of thing. If we don't know what that surface looks like, it becomes very difficult to simply take an experimental result and apply it to the real world. So let's think about this joint distribution of individuals. This is kind of like an epidemiological design. I'm going to follow individuals through time. They're going to be subjected to variation in the environment, some of it natural, some of it manipulated. I'll put this together with some hierarchical models and then sort of explore what this means for more aggregated variables that we care about, looking at sensitivities, inverse prediction. The design is a series of plots along climate gradients. So we've got Piedmont to southern Appalachians. There's climate variation between plots. There's variation within plots. So we've got a number of plots along moisture gradients, elevation gradients. So a large amount of variation spatially, temporally. And then we're going to also manipulate the environment. In this experiment, we can't manipulate climate, but we can manipulate the canopy. In other words, we can manipulate competition. So this sort of started back when I was young, and what we would do is to establish plots, let pre-treatment data accumulate for a while, and then implement canopy gaps and look at the responses. And so we've sort of done this periodically through time, set in 91, another in 99, another set in 2004, and just started some others. So when I give this talk 20 years from now, we'll be out here and I'll have good data on all of these. So what does the modeling look like? Well, most of the modeling of climate responses we do, that is on trees, the species distribution modeling, is based on spatial variation in temperature and moisture. And so one of the things I wanted to really understand from this is, does the spatial variation really represent how responses will occur to climate variation in the future? Can we sort of anticipate future responses based on the correlations we see in space? So the different plots, we follow climate through time. This happens to be winter temperatures, summer drought. Each plot has variation within a plot. There's variation between plots, so we can look at the average differences among sites and also the deviations from those average within sites. We can do that for both of these, and so we'll follow individuals on plots with plot level variation, temporal variation. We'll also have variation in the local environment, light and moisture, so in each location we have information on the light availability and moisture availability. So all of these things can go in with their interactions. This is state space modeling. We follow an individual eye. I'm probably not being assertive enough. Okay. There's an individual eye in year T. And so that's going to be represented by some states. These states are updated by demographic rates as they move through time. There's growth, there's fecundity, there's mortality. There's data models on these observations that help us to understand what these values are. There's error in the model. There's input variables that explain these demographic rates. And there's models for those data as well. And we use informative priors everywhere we can, and the things that we don't have information on are non-informative. So we use both. It's not either or. So just as an example of a submodel, maybe diameter and fecundity of an individual at time t, you could think of a vector of states. Those are involve maybe a vector of demographic rates. Let's say the diameter growth rate, the fecundity potential might be some vector y. Maybe x are our predictors. So in the basic core of this state space model, we can update this response vector based on the inputs x, some fixed effects and interactions, some individual effects I, some time effects t, some covariance matrix, so that we could sort of think about this as being the underlying process. As climate changes through time, here's winter temperature, summer drought. There are responses of every individual on all of the plots. So we've got a very high dimensional joint distribution of demographic rates. So this is kind of overwhelming. So I did all this work, right, and I modeled this thing, and it was even more complicated than when I started, right? So I've got more than the data. And so this is not where the analysis ends. This is actually where the analysis begins. It's not where it begins, but it's really where a lot of the work has to get done. Is you've got, you know, a very high dimensional posterior distribution. What does it mean? Well, now you really got to start the model to understand what this, you want this information. This is the information I've said we need, so what are we going to do with it? Well, through a series of papers, now we've been developing ways to actually turn this posterior into something that really helps us understand climate response. Here's one way. This is something I've called dynamic inverse prediction. And basically the idea is if we have a multivariate response, you could say an individual's health status is a multivariate response. There's a lot of variables, demographic rates, for example, associated with an individual. One variable is not a summary of the individual. You want to take all of the variables. Well, dynamic inverse prediction allows us to turn the model around and say, how does the multivariate response predict the input variables? And this is something that I thought was important, because if I want to know how important spring temperatures are for an individual, I don't want to look at the effect of spring temperature on fecundity, spring temperature and diameter growth on survival. I want to ask, how does that full response vector essentially predict that variable by inverting the model? Here's a species that's really good at predicting its environment. It can predict spring temperature. It can predict summer drought. This is the actual, so the two solid curves here are the actual summer drought and spring temperature experienced by these individuals. Here is the predictive distribution, which is a solid line and a credible interval. So pitch pine is able to tell you what the spring temperature is and what summer drought is based on its full demographic response. Other species have no idea what's going on. So here I've taken pitch pine again. I've just changed the scale. This is just the same plot I showed you the last time. Spring temperature and summer drought, very closely predicting both variables. Here's tulip poplar, has no idea what spring temperature is doing, and only kind of a little bit of a clue as to what summer drought is doing. Here's sweet gum, which is interesting because, again, it's clueless about spring temperature, but it knows what summer drought is doing. So we can do this for individuals, we can aggregate for the population and get a good idea of what variables are really important to individuals of each species. Here is two different species, individuals of two different species. Here's a maple, here's a pine. Again, looking at drought, this pine, again, is very good at predicting drought. This maple is pretty bad at it. What we want to do is get back from these demographic responses and relate them back to the physiology of individuals. This is work that Dave Bell has taken the lead on, and so Dave is over there. I'd point a laser at him, but I'd probably put his eye out. He's got sunglasses. If you could put your sunglasses on, I'll point this. Okay, so that's Dave. Dave is doing a postdoc at University of Wyoming, and he's been sort of the leader in the SAPFlux work here. What I wanted to point out is that here's a case where we can really relate the very close tracking at the demographic scale to how individuals respond to drought. So this happens to be a loblolly pine, but they're also very good at predicting the environment. Very strong responses to summer drought. Maple's not so good, and they're also very poor at the species scale. So here's a case where you could sort of say, well, okay, individuals and demographic rates, we can kind of see a connection between them. And that's the sort of relationship we want to understand better. If we look at winter temperatures, well, the pines can essentially respond to warmer winter temperatures. So when the hardwoods shut down in the fall, if it's warm in the winter, the pines keep going. So maybe that helps us explain why they do a very good job at tracking at the annual scale winter temperatures. And red maple, this sort of seems, well, okay, well, maybe on average it's doing pretty good, but the predictive interval is as big as a house here, so you don't even see the predictive interval. So it's actually getting kind of the mean okay, but it's just sort of, I don't know, the climate is out there somewhere, and who cares? Okay, so now how do we want to apply this to this practical application? The climate is changing. Can we help anticipate the kinds of responses we might expect? The predictions we have for the southeast are potential savannification, warmer, drier. We haven't seen change yet in the southeast in annual rates, but we have been seeing quite a lot of change in terms of the variability in precipitation. This is Wenhong Lee's paper in Climate, a Journal of Climate, and she sort of looked at this variation in precipitation through the past, and you're sort of going to get this wide swings, and that's really bad. If annual rates are really fluctuating a huge amount, it means you really get hit hard by these dry years, which are becoming quite a bit more frequently. And that can be tied to the dynamics of the Bermuda High off the East Coast, which is really responsible for our moisture supply onto the continent. So this is an effect that's happening now. And so the question is, does this sort of approach help us with understanding these kinds of responses? Well, what I wanted to look at then is, okay, let's try to think about these demographic rates that are being subjected to both longer growing seasons. That seems like a good thing for a lot of species. But at the same time, more intense droughts, that seems like a bad thing. And then there's this interaction with competition. So how are we going to put all of these things together and get to what individuals are doing and the output variables that we estimate? Can we go? Thank you. Okay, so this is one way to think about this problem. Did I lose something here? No. All right. One way to think about this is we've got, again, maybe a response vector y, and these could be demographic rates, could be diameter growth, could be fecundity. There might be a response r, another response r prime, a couple of responses in this vector. There's a number of predictors in our vector x. Maybe there's a predictor q, there's a predictor q prime. If we have those predictors in this model and we ignore the interactions, it's very easy to talk about the sensitivity of our output r to input q. It's just a. It's just the coefficient that maps r onto q, or q onto r rather. And so what we're assuming essentially is that there is an arrow going from every input to every output. Okay, now once we include interactions between inputs, and so we can put those interactions right in here the way we typically would, we end up with an individual effect here because the response to q depends on whatever q prime is for that individual. So in other words, the response to temperature depends on the light level that an individual is subjected to. So we start to bring in these interactions, and that makes this an individual level response. We've now just got arrows from the whole box to each of these response variables. We could go the other way. We could talk about interactions between the outputs, and so we could think about two input variables that jointly affect the entire vector here. And so now these output variables have connections, and that brings in the residual covariance. And so we've got the effect of Q on R here. We've got also the interaction with another response. R depends on R prime, and it depends on the covariance matrix here. So now we've got two arrows going to one response. But there's no reason to stop there. Why not look at all of the interactions where inputs can interact, outputs can interact, and so that becomes a bit messier. But now we're thinking about an integrated response of an individual to all of the ways these inputs can interact between themselves and how the outputs interact with each other. So in other words, we're trying to say there is an individual responding to its environment, not individual measurements on individuals responding to each input value. So we're trying to make this synthetic and think about it that way. Well, once we do that, we can look back then at the role of these interactions. So here's like the Frelich and Reich example. We could think about soil moisture status. Does it provide a refuge from summer drought? So you could think about as soil moisture declines, if these wet sites are buffered from drought, it means that the biggest response is going to occur on the driest sites. So there's a big arrow here. It's crossing many contours. At low moisture status on this axis, we have the biggest response to reduce soil moisture. That's essentially this negative interaction. However, if individuals respond to more moisture, but with more leaf area, increased evapotranspirative demand, we've got an interaction with competition. And when we've got an interaction with competition, it could go either way, right? Because as you dry down a moist site and it's fully utilizing the supply of moisture, then that could be really bad because you've sort of adapted to higher moisture conditions. And there's lots of examples of this happening. So all I'm doing is just sort of putting this in the context of how we interpret interactions. So this would be the case where you might actually get big responses on wet sites rather than dry sites. So either is possible. And what we find is lots of positive interactions. So we find an awful lot of cases where essentially the biggest responses to drought are occurring on the wettest sites, not the driest sites. Well, all of these interactions between climate variables, competition matter, and here's why we might care about this. So if we take a response of a species to winter temperature, which is on this axis, this sensitivity can be positive. Here's some positive values. Here's some negative values. And it can depend on the light level that the individual is exposed to. Well, this positive sensitivity means that we grow more when it's warm. That's why these values are positive here. That's what we'd expect for juvenile growth. We're seeing just the opposite for adult growth. Adult growth is showing a negative interaction. So in other words, when it's warm, the adults are not growing faster. If anything, they're tending to grow more slowly. Well, the reason that's occurring is due to an output interaction. There is an interaction between adult growth and fecundity. Fecundity has a strong response to winter temperatures. And that interaction between the two ends up being a negative prediction for growth of individuals as adults. Juveniles don't have fecundity by definition. They're not producing seeds. So we see a totally different response. Positive growth, negative growth interacting with positive fecundity. So this is an example where we've included both the input interactions, which are along these axes, and then the output interactions between adults, growth and fecundity. This is basically what I just said. This is what we expect, positive. This is not what we expect, negative. They look the opposite. And this trade-off between growth and fecundity would seem to provide an explanation for why that's the case. And also notice there is this strong interaction with light, so each of these responses to temperature does depend on the light level that you're at. Okay, so I think then as we start to apply this to responses to climate change, we can move from individuals to starting to think about landscapes, populations, and more aggregated variables. So this is an example. I've just put two plots on the same map. So here's a plot, here's a plot. This is a dry plot, a wet plot. And I've just put in symbols the responsiveness here in terms of the risk of mortality to summer drought. And what we can notice here is on the dry sites, we have a lot of individuals that really look quite sensitive to drought. So we've got some very big circles here, maybe not so much down on the wet site. So maybe this means that these loblollies, some of these guys on dry sites are kind of in trouble. They're at high risk. When we look at the responsiveness to summer drought in terms of growth rates, it doesn't look that way at all. We have certainly some are responsive here, but really down on this wet site, many of these individuals are quite responsive to drought, and it's not the same pattern we see for risk of mortality. So this is kind of a landscape level aggregate view that we can put together by aggregating essentially responses of individuals. If we sort of look at these in a marginal sense, that is, aggregate individual responses in terms of survival sensitivity to drought and the responsiveness, which is just another way of looking at that graph. We tend to find in general that the ones that are most sensitive tend to be most responsive in aggregate. So aggregated across populations, these are the guys that really respond a lot to summer drought in terms of this axis, and they tend to also be the ones that are sensitive. And then here's liquid ambar down here, which again is just kind of out of the game entirely. I guess it'll take over the world or something. Again, in aggregate, we can start to ask, well, okay, if we just aggregate everything together, we can look at a species level response. And this is, again, what we get from aggregating individuals. What I've plotted here are mature trees and juvenile trees. These are survival risk, and I've simply ranked them, their responses here to summer drought, which is red. And so you'll notice that some are quite sensitive to summer drought, some are not very sensitive. Well, what do these values mean? For a benchmark here, I plotted them with their responsiveness to light, right? So light is a risk that every tree faces every day, all the time. And so we could sort of think about, well, is summer drought a big risk relative to the other risks in your life, or is it quite a bit worse? And so what we find is for species on this end, summer drought is just as big of a deal as light competition, which is kind of interesting when you think about it, because light is killing trees all the time, right? They're just competing like crazy for light, self-thinning and all that. These guys out here, it's right up with light, and then out here it kind of goes off the scale, and so the sensitivity to summer drought is really not as big of a deal. Out here we've got a lot of the oaks, we've got some elms, we've got tilia, a lot of species that seem to be doing just fine, relatively insensitive to summer drought. Now this is a marginal view. This doesn't mean that every species that shows up marginally as being not terribly sensitive would not be sensitive in particular areas. We can marginalize in different ways. I've just here aggregated everything together. Okay, so where are we going from here? I think looking at individuals and their responses at this annual scale really does give us some perspective that's useful for understanding aggregate responses. And again, it's really sort of working from the individual up to the aggregate, analyzing at that individual scale, and then aggregating what we get in the ways that we want. But really the future that we're really working on now is trying now to really understand what is it about individuals that cause some to essentially help us do a very good job of predicting the annual rate from the very short-term responses in some cases and in others not. There's a lot of reasons why these two might not match up, and we want to know what those reasons are. So we're doing a lot more sort of physiology at this scale and trying to understand the extent to which it helps us determine what's going on at the annual scale. The other scale is we're working a lot with the FIA data now to try to see the extent to which these annual demographic rates of individuals can help us understand distribution and abundance. So this is a collaboration with Pankaj Agarwal in computer science, Alan Gelfand in stats. We've got a couple of postdocs, Suparno Ghosh, Thomas Mulhav, my PhD student Kai Zhu. We're looking at these climate gradients and 40,000 FIA plots and working on a number of ways to try to link demographic rates to abundance. This is an analysis we're kind of finishing up at the moment, looking at just sort of this classical idea of a climate niche. And we took a sort of a different approach on this. What we did is to decide to jointly model the seedlings and the trees together, but to do it in this conditional sense. So the seedlings depend on the trees being there and also on climate, and the trees depend on climate. And what I want to point out here is just that we don't get something that looks very Gaussian at all. So these are our climate niches. This happens to be southern red oak. This is for trees. Sorry, this is for seedlings, this is for trees. We get some very different climate envelopes for these two stages, and again, they don't look very Gaussian. They're kind of an interesting set of responses when you sort of take this view. So this is an example where we're sort of looking at these ontogenetic shifts in at least what you would model as a climate niche and trying to better understand what that might respond to. So taken together, I think this demographic approach is kind of a useful one, focusing on sort of an epidemiological approach to climate responses, weather responses of individuals, trying to connect it back to the scale where the physiology is happening, and then going the other direction to try to better understand what's happening at the continental scale. So if we sort of think about some takeaway messages from this, I think one of the things that I would really, sort of emphasize here is, if you can model an aggregate from an aggregate and do just fine, that's what you should do. There's no reason to get down into the weeds of all this detail if it's not necessary. There's a lot of cases where aggregate variables will predict aggregate quantities just fine. And I think if that's the case, you should do that. We've never been very good at scaling from one scale to another, and I think there's a pretty good reason for that, which is that this is a case where the process we care about really is at the scale of individuals, and it gets modified by interactions all the way up the chain that make for aggregate responses that really don't map very well onto the individuals. So I think this is a case where it's a good approach to get down to the individual scale. Think about modeling it at that scale and then thinking about aggregating to the scale we care about. The models are big, but they're actually not too complicated at least in a biological sense. I guess they're complicated in a modeling and a computational sense, but really these are really just really simple sorts of models. We're just conceiving of direct effects and interactions on demographic rates, and it's hard to get much simpler than that. But we are accommodating these kinds of interactions between the inputs, between the outputs. They happen at the individual scale, and then we've got to sort of think about how those interactions make for sort of these aggregate responses. So I like small, simple models. That's this third bullet here, and these kind of seem big and out of control. but really the idea is to get back to very, very simple quantities. It also means that when you fit a model like this, and you've got a very high dimensional posterior, I just wouldn't stop there. You just, you know, when you give us some credible intervals for some marginal quantities, and you say, well, okay, or you just give us some posterior distributions for fixed effects and interactions, you may be missing all of this rich information that's there. You want this information. It's not like these are all just nuisance parameters. You know, you got to estimate them because they're in the way, and so you just got to estimate them. We really do care about these intermediate quantities. This really is the point of hierarchical modeling. It's not just the marginal quantities we want. It's really what's happening at these lower stages that we want to know about. And so that means the modeling really can't stop with the posterior distribution. You've really got to start to think about how to put it all together. Okay, and then the last bullet I sort of beat to death already. The aggregation, if everything's aggregated before the analysis, of course, you're kind of stuck with whatever's information is available in the aggregate. So that's all I've got. And if anything was not crystal clear, I'd be happy to obfuscate further. So thanks for your time.
Theoretical implications of this kind of work, in the sense that in looking at, say, tropical polar storms, I could imagine a proponent of neutral theory saying, well, I can model that as stochastic commodity. Do you think that your approach of using the inverse prediction off of the framework of disentangling the stochastic demography and competition and the demography?
So what's your definition of stochastic?
I guess just by chance, you just randomly model it as opposed to being necessarily good.
What is chance? If I said, my mother died due to chance, what do I mean by that?
That's a good question.
Okay, so there's an explanation. Yeah, so one of the points I've made about neutral theory is when you put all the process into the stochastic parts, it doesn't mean that there's no process involved. It just means you're ignorant of the process. Yeah. And this is the big problem with neutral theory. And you see it when they compare neutral and niche models. I got dragged into this interaction because so I'm an editor on Tree, and a paper came in on neutral theory about the successes of neutral theory. And since I'm an editor, I said, well, go ahead and accept it, even though I think neutral theory is not very useful. Just because it's different than being a reviewer, I think. I think if you're an editor and there's a readership, you sort of say, you know, there's problems, but go ahead and print it. So anyway, the general editor said, okay, but would you write a response to it? And so eventually I agreed to do that, and that just came out in Tree. So you can look at this exchange if that sort of thing interests you. But when people take a neutral model and compare it to a niche model, and both are generated stochastically, and there's good examples of this in the literature, you get the same answer. You get the same dynamics. And the only difference is semantic. You've called one random niche generation. You've called the other stochastic recruitment. So either way, it's just success with different terms. So if you want me to say, how does this help us to test or not test neutral theory, my answer is it's not a concept that's testable. It's not a, you know, if you want to say species are the same, well, okay, maybe at some crude level, but if they have different dynamics and you want to understand those dynamics, you can't just say it was a coin toss. You've got to actually get into the model and say why. Why did the difference?
Yeah, I thought the tracking, the climate competition tracking is getting out there. I thought that was really cool.
Well, if you didn't put the climate variables in here, that's what you'd say. You'd say, oh, it's a neutral model. You know, that's the random variation. Well, what is the variation? Well, it's climate. And climate is the poster child for nonlinear dynamics. No climatologist says climate is random. It's highly nonlinear, and if you want to understand, some things are inherently unpredictable. It doesn't mean they're inherently stochastic. It means they're inherently unpredictable. I don't know. That's more than you wanted to hear. I'm sorry I answered that question. Somebody asked it. Yeah.
So presumably some of the variation in individuals is due to genetic variation. And so I was wondering what you think about the influence of evolutionary change and how that can affect the models to actually predict how plants will respond to climate change.
Yeah, so we know in trees there's a lot of genetic variation, and actually there's more in some species and less than others. We don't have genetic information in this model. I wish we knew more about that. But so what that's going to end up as in this model is individual level variation, because we're following individuals through time, and if there's a propensity for an individual to do more or less of one thing than another, it's in the individual effect. But that again, that's like the random part. It's really a statement of ignorance. So we can't, from this kind of modeling and these kinds of data, say what that contribution is. But there's no question that genetic variation is huge. In our warming experiments, I'll talk about tomorrow, we've made a big effort to include seed sources from different locations, and we do analyze those. And this is well known. So obviously you're completely right, and this approach won't do everything. Yeah.
I guess maybe this follows on a bit from that last question, but the results you showed on the individual sensitivity and the responsiveness to different environmental conditions, those were for stands that were within one portion of the geographic range of species that you're studying.
So
I guess I'm wondering if you would possibly expect there to be different kinds of responses in, say, different ends of the species geographic ranges.
yeah, so we just started a project. This is through the Macrosystems program, where we're initiating sites from the Northeast, the Upper Midwest, down through Florida and Puerto Rico to look at variation. And a lot of the species are well distributed across that entire range, so we have a lot of latitudinal variation. But even for species that aren't well distributed latitudinally and longitudinally for that matter, we tend to have quite a bit of variation within sites in terms of elevation and climate variation and all these other things. So we do hope to get a very rich sense of how responses. Actually, it reminds me of the conversation that we had this morning, where you were talking about looking for that same sort of variation in streams, and I think we need to certainly do that as well. We have looked at microsatellites of trees and seedlings in some of our plots. This is a student who recently finished, Emily Moran, and that actually was very helpful in helping us to see the genetic variation in reproductive output, which turns out was higher than we predicted by the demographic modeling. We found that reproduction was more concentrated in fewer individuals than we thought, even though we already thought it was pretty highly concentrated. And so if we get more of that kind of work done, we'll start to be able to also look at the variation in terms of responses to climate variation.
Okay. So given what we know about the genetic variation between species, there's very low genetic variation, and I don't really know much about the other species. Especially like this, does that map a little bit well to how patterns of aggregate predictability seems to work? Like finds fish pine work really well. Is there some circumstantial evidence that suggests that those ones that have lower genetic variation?
Well, that would be interesting if it did. There's no reason why. The question was very insightful, and I forget what it was. Do the species that track climate very well. Are the individuals, is there lower genetic variation in those species? Is that? Yeah, okay. So that's certainly possible, but it's not necessarily required. It could be that, you know, I have no idea what it is in pitch pine. I don't know what the variation is. We know a lot about loblolly because it's a commercial species, but it wouldn't have to be the case. That variation could be, the species that are highly variable, it could be because they're really reacting to more variables in more complex ways, I guess. Yeah.
I'm curious in your inverse modeling approach, because that turns, I think, sort of the traditional notion of, you know, cause and effect, in a sense, on its head. I wonder, just as sort of a general philosophical point, if you would speak to how you can use that approach as sort of using the I guess, the posterior to, you know, model the input variables, how that can serve a sort of in a predictive, what are the obstacles or the limits to that serving as a predictive model?
Yeah. Well, I think the notion of using biology to predict the abiotic environment is not new. I mean, just kind of, you know, this idea of weighted averaging, where you get a bunch of species, you know their environmental tolerances, and you come up with some index for a plot. You know, in a sense, that's kind of taking a multivariate biological vector and saying something about the environment from it. In this sort of dynamic case, I was more interested in it as a way to get some sort of integrated index of how important that variable is, because I don't know how to do it the other way. If I take X and I say how well does it predict all three response variables, for example, if there's three response variables, I don't know how to weight that response. Is the first response important to get right and the other two not so important? I'm not sure how to do that. But if I go the other direction, I'm simply using the model. That's all I've done is I've just inverted the model. So if I think the model makes sense in a forward direction, if I just turn it around, I'm not sort of trying to take the output of the model and then say, you know, what does that say about the importance of X? So I'm sort of viewing it as to me, it seems like a really useful way to integrate the, or at least gauge the importance of the environment.
That makes perfect sense. I guess what I'm wondering is, can you take that and use it in a predictive framework to, maybe I missed that.
Well, the model itself is, can be used in a predictive mode. And we've done some of those kinds of things. You know, I've sort of so far been trying to really understand what it tells us about the responses to the environment now. But with Pankaj Agarwal, we have a big stand simulator we've been working on for years to put all of these estimates into a stand simulator, and it's been very hard. It's very hard to get a good predictive model because of the nonlinearities that are involved. But that's something we've been working on. I'm sorry, even if there's no more questions, I'm not going to let you, no, okay. I guess we're done. So thank you very much. Oh.